BIN310 Introduction. So, first I want to say a few words about microbial genomics, but because that's what we are going to do in this course. Selected topics in microbial genomics is its name, right? So let me just first say a few words about this. The world has changed. I see it in the water. I feel it in the earth. I smell it in the air. So where are these sentences taken from? Well, you may recognize these are actually the opening words of Lord of the Rings. And the reason I use these sentences here is because when we are talking about the selected topics of microbial genomics, then indeed the world has changed. We sequence it in the water, we sequence it in the earth, and we sequence it in the air. Because this is exactly what we are doing in modern microbial genomics. We sample the microbial world from all spheres of this planet, the water, the earth, the air, and also from ourselves, or animals, or plants, or fish, or whatever, insects. This we collect, we do some library preparations in the lab, and then we do some sequencing in order to reveal the DNA sequences of whatever is found living in these environments here. Now this is what we call metagenomics. And this is sort of a branch, you could say, of the genomics in general. But it's quite important, at least nowadays. I also want to emphasize that microbes, despite what you might get impression of when you see, for instance, TV programs about life on Earth, you never hear about microbes, do you? You only hear about elephants and whales and people, right? Imagine a handball court. You all know what a handball court is, right? The field where you play handball. This is approximately 40 meters long. Now imagine each centimeter along this handball court corresponds to 1 million years. And let's look at the history of life on this planet. Picture as distance on this handball court. The microbes, they have been around all the time, since the dawn of life, 3,800 million years ago, which corresponds to 38 of the 40 meters here, right? And they are still here today, very much so. The eukaryotes that we are part of, the multicellular organisms, or organisms with the core nucleus, they have been around only for 15 of these meters, right? Around here somewhere, roughly. This is of course pretty rough numbers, but no one was there to actually observe it. The fishes, at least the what we today call fishes, they have been around for like 500-600 million years. So the last part here somewhere, right? The land plants, a little bit less, 4 to 5 meters, and the insects, 3 to 4 meters. The last part here only. The reason I mention insects is that our insect professor at NMBU, she wrote a book called The Planet of the Insects. Well, It's actually not the insects that are the sort of primary biology of this planet, as we can see here. It's the microbes. And for humans, we have been around for the last 2 millimeters of this umbilical cord. So close to nothing, right? It's tempting these days, since we are coming close to an election, to sort of imagine what does it look like if we step 1 meter further into the future here. 1 meter. That's 100 million years. The microbes are still here, I'm sure. I'm not sure about the rest of this. Of course, the microbes, having been around such a long time, they have evolved into a huge diversity. And they are found everywhere, basically. From the deep sea sediments to the clouds above us, and on us and inside us, and basically all other living organisms of some size are populated with microbes. So they are everywhere. This is the primary life form of this planet. Here's another picture showing the same thing really, but in a different way. So this is a tree of life, a phylogenetic tree, and at each end of the branches here you find some group of organisms, right? And the length of the branches indicate how far they are apart in terms of evolution or evolutionary distance. And here we can see the 3 main domains of life. This is the bacteria up here, the big one. They are all microbes, right? So down here we have the archaea, this branch here. They are also microbes. And here the green branch here are the eukaryotes. And this phylum here, Ophistoconta, that's where we belong, the humans, along with all other animals and insects and fishes. And you can see that the diversity among these animals, fishes, insects, is not very big if you compare to the diversity between the various bacteria. It's an enormous diversity, which is not so strange given the previous slide where these have had a lot of time to evolve, right? While we have only been around for a very short time and haven't managed to evolve very much yet from each other, right? From each other in these branches. There is also another interesting facet of this picture here, and that's what I call the biological dark matter. All the branches that has a red dot are branches where we lack the ability to isolate and sequence the representative from this branch. And you can see there are lots of red dots here from the bacteria and the archaea, none of them in the eukaryotes. And this rather dark branch on the right hand side here are all taxa that we did not know existed before we started doing metagenomics. These have simply been around all the time and we never knew they were there until we started metagenome sequencing, like 10-15 years ago. Entire branch here, completely new discovery. And last year I saw this publication here in Microbiome trying to estimate what have we still not seen, right? Because we have just now started really digging. And they conclude that so far we have probably seen around 2% of the global prokaryotic taxa. So it means that this big branches here still only represent 2%. There are 98% left to discover. And this tells us that, again, it's a planet of the microbes. Metagenomics, what is it really? Well, It's based on the very efficient DNA sequencing technologies we have today. So it's all about sequencing DNA, piecewise reading of the DNA. And it means we collect whatever lives inside the different environments. We do some filtering and processing and lab preparation, extract all the DNA. We don't know which organisms are there, but we know they all have DNA, right? And then we do sequencing to get some data that we are going to work on. And these are the type of data we are going to work on in this course. Sequencing data from microbes, because this is almost always only microbes. If you collect all the DNA in an environment, it's almost exclusively microbes. If you sort of took a human being, a whole body of a living human being, and squashed it through here, extracted all the DNA in a human being, and sequenced it, and counted the number of genes, you will find that number of genes coming from the human is like 1%, and the genes coming from all the bacteria living inside this human is like 99%. So we sequence all variants of a gene in an environment, and that's for marker sequencing, which is not something we are going to talk a lot about in this course this year. We are instead focusing on what we call shotgun sequencing, which means we sequence all DNA. That's the basic. That's genomics, I would say, in general. This marker gene sequencing is not really genomics. It's also very, very useful. But I think this year we will not talk very much about it. It has to do, we can't cover absolutely everything. So what we see with these eyes is this, right? Lots and lots and lots of short or not so short DNA sequences. And that's where bioinformatics comes into the arena, right? We have to do some bioinformatics to get something out of this. Massive amounts of DNA sequences. Let me just say a few words about the relevance of microbial genomics in a sort of NMBU perspective. Why do we do microbial genomics at NMBU, and for what reasons, right? First of all, of course, we have microbiologists who are very good at studying the basic biology, you could say, of microbes in general. And of course, it's difficult to study microbes without having some element of microbial genomics in the projects. You can do a lot without genomics, of course, but it's also sort of a natural part of microbiology. Of course, we also do a lot of medical relevant studies at NMBU, both human medicine and of course veterinary medicine. And in both cases, microbial genomics is important. For instance, for tracing pathogens, in whether they are in hospitals or in farm animals or whatever. Studying the interaction between the microbes and the human body. That's a big topic, and we are also doing that at KBM. And also, of course, this antibiotic resistance problem that you have all heard of. And microbial genomics is also important for that type of studies. Food production and food safety is also a big issue at NMBU. And of course, both in the production of certain food items, microbes are used in the production, but they are also sometimes problematic, right? In the sense that they may compromise the food safety of either during production or afterwards. And we have all heard about this scandals, you could call it, where people are getting really ill from food poisoning, and that's due to microbes, right? So tracking pathogens and and helping food production is also a topic where microbial genomics is used quite a lot, both at KBM and our sort of neighbor institute, the Veterinary Institute and NOFIMA, the Food Research Institute. They do a lot of microbial genomics, both of them. And of course, the interactions between the production fish or animals or plants and the microbiota. That means the microbes that live inside them or on them, on their skin or whatever. Salmon breeding and farming is of course a big topic at NMBU. And salmon feed production. Also other farm animals and plants. All of them have interaction with microbes, and in all these cases you will find research activities at NMBU that study the sort of the interaction between these animals or fish or plants and the microbes. Also, environmental studies are using microbes these days. It's used as a sort of an indicator, right, of an environmental status. As an example, we have a project starting right now at KBM where we are using the microbial composition of sea floor sediments to categorize the pollution, you could say, from fish farming. But also water quality and things like this are characterized by not necessarily the amount of microbes, but the composition. And that's something we will talk about towards the end of this course, actually. Forensics, yeah, that's not something you typically think of at NMBU. But actually we have been involved in quite some forensic projects, both in national and internationally in the last recent years, where we use microbial genomics in various ways. Also in forensics, or in order to say something about traces after crime, crimes that has been committed. And even anti-terror surveillance, the Defense Research Institute in Norway, we have had some collaboration with them, where we simply sequence the microbes you find in the air in the subway in Oslo, in order to detect something that should not be there, right? And there are, of course, many other applications at NMBU that I am not aware of, and new are coming all the time, involving microbial genomics in some way or another. So, the selected topics in microbial genomics, this course, what is it all about? Well, the world has changed in more than one way. Because if I look back 10 years ago, this course having the same code but a slightly different name, it was all about the models and the algorithms in bioinformatics. So it was a much more theoretical course back then, looking at the theory behind the methods. But this has also evolved, right? 10 years of evolution in microbial genomics. And today, this year's version is quite different. And it's more what I call pipeline science. It's sort of a phrase I have invented myself, and it's not really, I invented this in a sort of a dark hour. This is not meant to be a positive statement, really, but it's not that bad. The point is that this field has evolved, going from this more theoretical sort of way of approaching it to a more practical. That's what I mean by pipeline science. So this is not only because the world has changed, I have also changed. As a teacher or pedagogical person, you could say, my philosophy, I have found that in order to learn tools, and that's what we do here, right? Whether it is in statistics or informatics or bioinformatics or whatever, computer science, right? You learn how to use the tools, then it's always, I have found, better to start by learning how to use the tool first, and then afterwards you start digging into how it really works. Because when I was a student, it was the other way around. Then the professors told us how it actually works, but we never knew how to actually make it work, or why it worked, or for whatever reason we were going to make it work. Only focus on how it works. And I found that it took me a lot of extra time and effort to really understand this when you get it that way. So I think it's much better first start using a tool and then learn how it works, if you need to, because of course in many cases you don't really need to learn a lot about how it works. You can still use it. Think of driving a car. You don't need to know exactly how the car works in order to be able to driving it. And also the computers we use every day, who can actually tell me they know exactly how it works inside? What are all the details going on inside your computer? You never know, really, but you can still use it. It's a useful tool, right? So I think this is sort of a pedagogical philosophy I have obtained over the years, and seen that I think this is the best way of learning these type of stuff, actually. So what we are doing in this course now is to focus on tools, and I should also stress that I try to focus on the more basic data processing, right? And what do I mean? I mean that we have the raw data coming directly from the sequencing machines, the basic processing steps that all people who are going to do this need to do. And we try to cover wide instead of digging deep here now. So we will cover quite a number of tools, but we will not dig very deep into any of them. And we do not dig into the specific biological questions, because that would mean we have to stop right there, right? And spend a lot of time on that biological question. So instead of digging deep, we cover wide this time, or actually I've done so for all the time. Actually, I'm not a biologist myself, so in order to dig deep into the biology, it would mean I could only do that by doing a lot of efforts, and also I'm not sure it would be very good. And also I think that in order to be sort of useful to as many as possible, a course like this should not dig into one particular biological question, but rather try to cover a lot of topics and learn how to use the tools. And that's what I talk about here in my I think this is my last slide in this first session, the microbial genomics keyboard. So learning programming, I hope and expect that you have all learned some programming in R or Python or whatever. Learning programming has some resemblance to playing music, in the sense that you first have to learn the keys on the keyboard, right? The coding elements. How do you actually spell the syntax, right, in the coding? What elements do we have, right? We have objects, we have functions, we have loops, we have all these coding elements, right? And how to sort of pronounce them in the different languages, but they are the same elements, right? And then once you have learned this, you start to combine them. And that's when you start to play the music, right? Making the programs. So in the first part here, you play with one finger on the keyboard, right? One tone at a time. But once you start mastering this, you can really start to make music out of it, right? By playing all the elements together. And this is also, I think, applies to this pipeline science type of working. And we can see it as some kind of meta programming, actually. So we now try to learn a long range of software tools. Well, it's not that long, but at least it's a range of software tools. Those are the keys, right? And once you have knowledge of all these tools and how to use them, what are the inputs they require, what will they give for output, what do they actually do, right? Then you can start combining them and release the full potential of these tools when they start to play together, right? And that's what I'm hoping that we will try to. We will not actually accomplish that in this course, but at least I hope we can overcome the first obstacles. Because in order to become a musician, right, a bioinformatician, right, you have to know the software tools, and that of course takes some time to learn. There is no shortcut here. It's just like in learning a programming language. I mean, if you have, for instance, have learned R, it's not a big effort in a sense to learn Python, because the ideas are the same, but it's still a big work because you need to know all the new words, right? And the same here. You need to know all the tools. That's the words, right? And of course, in addition to the tools, you need to be fluent in UNIX shell, which is sort of a coding language, plus some more sort of standard programming languages like R or Python. In this course, we will use R, at least I will. And all the examples and everything is in R, nothing in Python. I don't use Python very much, so that's the only reason. We could easily have done it with Python as well. UNIX shell is something we're going to look a little bit into in this course, not super much, but only enough to manage, I would say. And we will look into a number of tools. I'm not sure how many, but 20, 30, something like that we will see during the course. Well, we also need some practical information in this first introduction in order to get started here. First of all, the weekly calendar. How will the weekdays look like in this course? First, the pedagogical philosophy is flipped classroom. I guess you have experienced this in other courses before. And what does it mean? It means that in Canvas, I will add basically every week a new module. And a module consists of an HTML file with a lot of text containing code example. It contains some descriptions of what we're doing and why we're doing it, blah, blah, blah. Code examples, links to some lectures from time to time, or articles or websites or whatever I would like to point out to you, and some exercises here and there. You work on this on your own, and I strongly encourage you to form groups, working together in groups, whether you meet physically or only by screen. It doesn't matter to me. But work together if possible. I think that's a very big advantage if you can do that. It's so much faster to learn stuff if you have someone to discuss with. If you have problems or questions or whatever, we meet and discuss this, and that's when that's sort of my role in this is only here, right? As a teacher, I am not going to give lectures in the classical way, because my lectures will be as video links, just like the one you are watching now. I don't waste time doing lecturing. The one-way lecturing is a waste of time. I feel I can just make a video like this, and that's it. When the valuable time that we have for two-way communication, we should use for something more useful, and that's what I'm hoping here. That you work on this on your own, and then you come and ask, and we can have discussions and try to solve problems together. And where do we meet and how do we meet? Well, we have these times. These have been allocated for us by an MBU system, and I'm not going to change it because I know that this has been designed to not overlap with some other courses that are compulsory to some of you. So trying to change this schedule here is hopeless. I have tried it before. These are the 6 weekly hours we have, from 8 to 10 on Thursday mornings, and then from 12 to 4 in Friday afternoons, which is of course a little bit suboptimal in many ways, but that's the way it is. So all this time is reserved for questions, discussions, and problem solving. And you can show up whenever you like, or if you need to, in these hours here. There's nothing compulsory about it. It's just a service, you could say. Due to the infection control, we are still not done with the pandemic. And I mean, bioinformatics is a topic that easily can be taught by screen, right? Some courses cannot do that, and they will have to have physical meeting. We don't have to, and then we should not do it either, at least the way the situation is right now. So we will only meet on Zoom. I will put out some links on Canvas every week where you can come to the Zoom meeting and talk to me or someone else. This physical meeting, we may adjust it. And start meeting physically, because we have the rooms reserved. And if let me put it like this, if the pandemic situation allows it and there are enough of people who want to do it, we can do it. For me, it makes no difference. But I can only do either one or the other. I cannot do both physical meeting and Zoom meeting at the same time. That's not possible. So we have to choose one or the other. And I have a feeling that most people will choose the Zoom approach, right? But we can combine it in the sense that we can say that, okay, some of these hours are for physical meeting, some of them are for digital meeting. But we will start out by having only digital. So we will see how it turns out. There is one exception though, and that's the first day, and that I will come back to. And I have to stress this. We have this year put a limitation to the number of students, and this was blown sky high immediately. We are now twice as many students, more than twice as many students as it has ever been before. This has simply exploded, the number of students registered for this course. So you have to be patient. We are not really, we are not prepared for this many students coming to this course. And it's not like we can just immediately allocate more people to this, because there are no more people. Everyone else are busy doing something else, and also you have to have the proper background, right? So I have been lucky that one PhD student at our faculty has said she can come and help me out a little bit. She has done this before and is a very excellent student and she also has some experience in being a teacher. So let's hope that will be. But I say you have to be patient. It could be that these Zoom meetings will be overcrowded at times, right? But that's because we are too many really on this course now. But we will have to try to manage. I think it's almost impossible to deny people coming, learning something when you are at the university. Universities should not deny people learning. The exam. There is no exam, and that's new this year. Instead, we have these compulsory assignments, and I have made 5 of them. I should say I have only made 5 of them because we are so many students. I don't think we can have more than 5 compulsory assignments. Ideally, I would like to have a little bit more, almost every week, but this is what we do now. You have to pass all these assignments to pass the course, and there is no grades, there is only pass or fail. So you should immediately start working on this once they are launched. They will be launched parallel to the relevant modules. So the first assignment will be launched immediately, almost the first week, because it's based on not on what you learn here, but what you should learn before you come here, really. But that's something we can, you will find it on Canvas. So how does this work, this assignment? Well, you make some short reports in R Markdown. We will come back to how to do that because you may not have no experience in how to make R Markdown documents in R. It's very easy. It's very easy, I must say. So don't be afraid if you haven't done it before. You will do this quickly. You hand them in on Canvas just like you would otherwise do. And I noticed that there is in the description of the course it says that there will be some oral presentations. No, there will not, because we are simply too many people. We have no time for doing this year. So it will be only a written. You hand in these written reports in Canvas, and that's it. There is one deadline for all the projects, really, or all the assignments. So by Friday, December the 3rd, which is the last day of the semester, all of them must be approved, right? So you can basically hand in any time during the autumn, but make sure you have gotten it approved in good time before the semester ends. So you should hand them in as we go along. Don't try to sort of wait until the last minute here. So, and if you hand it in good time, you can get some second chance also if you did not get it approved, in case there were some crucial elements lacking and it wasn't approved. Then it's a good idea to get this feedback in good time, so you can fix it and then hand it in again, right? If you wait until the last second, you will not get this chance. So you cannot expect to do all these 5 assignments quickly in the last afternoon. That's a really stupid strategy, I would say. High performance computing. That's quite an important element of this course. So you will all get access to our local computing cluster Orion. And that's something we will talk about later. We will do all the coding and computation on this Orion cluster. So you will not use your own computers for computation. You will use your computers, but only as a sort of a interactive communication to Orion. And it's not that the things we are doing in this course are so huge or demand such a enormous computational power that we couldn't do it on a laptop. In many cases we could have done it locally, but we want to do it on the Orion because it's part of learning here, right? UNIX cluster computing is standard in all computing sciences, I would say, also biological sciences. So it's one of the learning goals to be familiar with how to work on these high performance computing facilities. In order to access the Orion, you need a VPN connection, and that's something you have to fix on your own computer now, before the course starts really, or before we really start accessing the Orion. So this is your first sort of homework. Make sure you have a VPN connection, and I'm not. This is not part of my job to fix this for you. This is part of the IT department. They have made some documents that you can click into, and if you don't manage, you should talk to the IT department or whatever students talk to when they have computer problems, right? Because this is not sort of bioinformatics. This is pure informatics. But I have some links here for Windows or Mac users. It shouldn't be that difficult. At least I managed. If you are a Windows user, you also need a terminal app, or everyone needs a terminal app in order to log on to the Orion. But for Windows computers, you have to install some program, either this MOBA Xterm or PuTTY. And you can install them from the software center, or you can just download them on your own directly from the internet. They are very small and easy to install programs. I think I will use the first one here. But the second one, personally, I use both. But they do the same thing, so you only need one of them, right? You will see on the first module 1, description how to do this and the links and everything. For Mac users, you don't have to install anything because Mac operating system is already a UNIX system and should have the app called Terminal with a capital T. That's all you need really on a Mac. So the Mac users have a easier in entrance here, really. And how do you access the Orion? Well, first you have to get a user account. So everyone gets a user account, which means you get a username, and the usernames are all like this. They start with BIN310, then this hyphen, and then a number 1, 2, 3, 4, 5, 6, 7 up. I don't remember how long, but we should have plenty of usernames, I think. You also need a password, of course, and you will then get what we call a dummy password. So when you get your username, you also get a dummy password that belongs to that username. And the very first thing you do is to log in using that username and the dummy password. And then you have to immediately change to a proper password, right? Proper password. And I think the IT people has set it up so that this is the first thing you have to do. But I will also show you how to do it. So you have to construct your own proper password, right? So that this dummy password is no longer in effect. This is of course for safety reasons, computer safety. And also I cannot send the passwords by email. That's also part of the security here. So it means that in order to give you the passwords, I suggest that on the very first day of this semester, or at least for this course, the first Thursday, I think it's the 9th of September, 8:15 in the morning to 10, then we meet physically in this room here, BT3810. This is the one we have been allocated for the Thursday mornings. And then I will give you your username and your password physically there. You can show up anytime in this. You don't have to come at 8:15. It's actually nice if not everyone comes at the same time. But during this time, this morning, right, from 8 to 10, show up, get the password, and then you can leave again if you like, right? Or you can sit down and just log in and be done with it, changing your password. You decide. But anyway, if you for some reason cannot show up physically, then you must send me an email with your phone number. Because then you will get an SMS with your username and dummy password. We cannot send it by email, but SMS is safe. There will be some messages for this on Canvas as well. Let me also say a few words about the prerequisites of this course, and this is taken from the study handbook, right? So we expect you to have some introductory course in bioinformatics. So you should know something about sequencing, some basics about sequence alignments, and a little bit of blasting and these type of things. And also R coding is part of BIN310 a little bit. Statistics, yeah, the Stat 100. It's not that we need a lot of statistics in this course, but it's always nice to have these. What is a stochastic variable, right? These type of thing, conditional probabilities and stuff. So you don't have to sort of sit and wonder what I'm saying, right? It's pretty basic things, but it's also nice to have these basic things in your head. And then, of course, some basic programming. I think STIN 100 is R programming, but it doesn't have to be R programming. Even if we are going to use R in this course, and I'm not going to be teaching R in this course, but if you know programming in Python, for instance, very well, it shouldn't take very much effort to learn enough R to follow what I'm doing. The important thing is that you know the sort of the concepts, right? Data types and data structures like your vectors, matrices, tables, lists, and looping and functions and stuff. If this is completely alien words to you, then you have a little bit of a problem here, I think, because this is something we're going to talk about and not really teaching. So you have to do a lot of catching up on this then, at least. So there will be no advanced coding required. It's quite simple stuff that we are going to do. And all the coding will be in R. And we will also make shell scripts, and that's really basic stuff. So because we don't expect anyone to know anything about shell scripting. And if you do, it will be, you will see that this is pretty basic stuff. But R coding, a little bit more than the very most basic then. So RStudio, we will use RStudio on the cluster. You will not need RStudio on your local computer, because we will do it also on the Orion computer. And we use RStudio basically as an editor to edit and inspect all kinds of files, not only R files. We make some R scripts, of course, and we also make some R markdown documents, or you make some R markdown documents, and that's the assignment. We will not have time to teach R coding. So in case you need to sort of refresh or learn some R coding, you will have to do it on your own. I will not have time to teach you that. But there are endless sources online for learning R. And if you look at I think in module 1, I have linked to the BIN310 lectures and stuff. You may pick up something there as well. The UNIX shell, that's completely new. So in modern biology, we must be familiar, at least to some degree, with this UNIX command line, because that's what you meet everywhere in computational sciences. There are many family members, you could say, in this UNIX operating system family. One of them is Linux. And actually, Linux is a variant of Unix that we are using on Orion. Actually, I saw the other day it's a 30-year anniversary for Linux this year, or actually this month, because it was invented by a Finnish guy, Linus Torvalds, exactly 30 years ago, 1991, in August 1991. So that's the operating system that we are going to use. Those of you who use a Mac already use a Unix system without knowing it, I suppose. But this is also a Unix system, but Windows is not. And the Unix is based on a file tree, you could say. The whole operating system is built around this file tree, which is a hierarchical structure of directories or folders, right? So the top of the tree is this folder here called slash. It is a slash, right? And this is actually the name of a folder, a directory, right? Under this, you find a number of other folders, and this is only some of them. They typically have these short cryptical names. And under the MNT folder, you typically find some other folders, and one of them is users. And then under the user folder, you find all our home folders. So this is my username on Orion, and these are yours, right? There will be many of you, many users, and they are all under this folder here. So when you log in to Orion, you always log into your home folder, whatever. If your username is this one, you end up here in this folder, /mnt users BIN310. Yeah, we will see this a lot. We need to know enough UNIX commands and shell coding to maneuver up and down in the file tree, of course, handling files by copying them, deleting them, moving them, inspecting them, and etc. And we have no graphical interface. At least we have to some degree in RStudio, but the point is to learn to do this without the graphical interface, because then we are not dependent on it. And that's a big change for some people who have never seen a command line before and worked on a terminal window. So that will be a new experience, but it's valuable, I would say. You will guaranteed meeting it if you are doing bioinformatics later in a more professional way. And we will make some simple shell scripts, and that's it really for the Unix coding. It's not very deep. We're not going to dig deep into it.